I'm not usually fast with these videos, but I'm going to try to be quicker this time. We're going to talk about theoretical applications of biologics because really that's in the realm that we're in. There are a lot of potential applications for biologics that aren't being used, that aren't being researched, that aren't being investigated because of some of the limitations with regard to the way we can use and the way we can study biologics through the FDA. Right now, there are two kind of tracks. You've got a drug track, which is Section 351, and you've got a human cell and tissue track, which is Section 361. And if we're talking about the framework that is neuropathy, neuropathy is a disease. And because it's a disease, we're not using human cell and tissue products for this process. We can only use drugs. Now, the current drugs that we use for neuropathy, most people are familiar with. Gabapentin, Lyrica, tricyclics, CBD maybe, NSAIDs. We have some supplements, alpha-lipoic acid, even nitric oxide. If any of you know Nathan Bryan, he's one of the foremost researchers on nitric oxide. He's got patents. He has some marvelous research on the application of nitric oxide in neuropathy, but that's still considered a supplement. Theoretically, in my opinion, there's the potential to use biologics, not in neuropathy, because again, neuropathy is a disease, but theoretically, could you apply a human cell and tissue product in a patient with a neuropraxic deficit, a collagen-based deficit of the peripheral nerve? Theoretically, you could. Now, if there are some loosening of FDA restrictions and we're able to do more investigational studies, not just observational studies, not just these patients had this done and we observed this happened, but if we could actually do randomized trials, if we could do comparative trials, those currently are drug studies because there's a null hypothesis. And the null hypothesis is using XYZ, right, say PRP, to treat a disease process or even a collagen-based deficit in the peripheral nerve. If you use it to treat that paradigm, you're imparting a biologic activity on that human cell and tissue product, which renders that study a drug study. And this is why any randomized controlled trials that you would see that are out there that discuss the use of, say, PRP or BMAC or something else to treat a disease, whether it's neuropathy or osteoarthritis, those are really drug studies. Were the FDA to loosen some of their investigational roadblocks, and we could do comparative studies, it would be absolutely fascinating to know. Well, we know that gabapentin can work in some patients with neuropathy, but there are lots of systemic side effects and most people don't tolerate that product, that drug. Well, if we did a randomized comparative trial, a drug trial, and we were able to see, well, does gabapentin work better than the focal application of a biologic, a human cell and tissue product, for a cartilage-based defect in a peripheral nerve? And we did objective measures. We did nerve conduction velocity testing, or we did monofilament testing, both objective measures. We could potentially determine which is better. Would there be value in that? I submit there would be tremendous value. Why? Well, right now in the United States, neuropathy may not seem like it's a big deal, but close to 46% of the US population is diabetic or pre-diabetic. Around 30% of those patients suffer from neuropathy. That means that conservatively 30 million patients in the United States are suffering with this disease process. And that disease process exists on a spectrum. Some patients may have neuropraxic deficits. They just have an issue with the outer third of the nerve, the epineurium. It's giving them numbness and tingling. Their disease process hasn't progressed to the point that there's damage to the outer third of the nerve the middle third of the nerve and the inner third of the nerve, and now that nerve is dying off. We'll review just a touch of anatomy, all right? The nerve is wonderful. It's kind of like speaker wire. You've got the little cable fibers in the middle, but then you have all this insulation around it. You have the endoneurium innermost, perineurium in the middle, the epineurium outside. Now the epineurium, interestingly, is dense irregular tissue. Histologically, it's type 1 collagen and type 3 collagen. Fascinating. Myelin heaps up in mounds on that structural scaffold. And myelin only attaches to the epineurium because myelin uniquely must attach to an extracellular matrix product. And the epineurium is that. It is dense irregular tissue. It's got structural scaffolds. Now, flow down the nerve happens by electrical impulses, axoplasmic flow, jumping from little spaces in between the myelin. That's called saltatory conduction. That would be like driving from Las Vegas to LA and jumping every 4 cars. It wouldn't take 4.5 hours, maybe take an hour. How fabulous would that be? No need to build the bullet train. Well, in early neuropathy, what we find if you do nerve conduction velocity testing, and in particular, if you do something called inching, which is very painful for the patient, but repeated shocks can give you definitive information about where specific compressive regions in the body might be causing cartilage-based defects in the nerve, across the carpal tunnel, across Guyon's canal, across the ulnar nerve, across the fibular head, across the tarsal tunnel. These are all regions of potential focal nerve entrapment that would lead to cartilage-based defects in the nerve. Theoretically, because the epineurium is dense irregular tissue, type 1 collagen, type 3 collagen, could you use a human cell and tissue product to supplement missing, damaged, deranged, deteriorated, dense irregular tissue? Theoretically, you could. You could apply something like PRP or BMAC, which has type 1 collagen. I would submit that an allogenic allograft like Wharton's jelly might be more robust. It has both type 1 collagen and type 3 collagen. You tend to get more comprehensive collagenic differentiation. Now, this is, of course, theoretical because we haven't had the liberty to do randomized, controlled, prospective trials. Have there been any observational studies? Published? Not yet. But am I aware of a group that is doing that observational work and they've collected over 100 patients with neuropraxic deficits as defined by nerve conduction velocity and monofilament testing? Yes. And they've applied biologics, Wharton's jelly in particular, and have they found significant improvements in the patient's symptoms, numbness, tingling, proprioception, and in particular, any improvement in nerve conduction velocity testing or monofilament testing? As a matter of fact, they have substantial improvements, 40% improvement in nerve conduction velocity testing and monofilament testing. That's objective. That's significant. Will we see a randomized controlled study or a comparative study to say gabapentin or Lyrica or tricyclics? God willing, with this new administration and maybe some loosening of the FDA regulations, we might be able to do more investigational work. Because ultimately, if we're going to make the greatest impact on health, if we're going to make an investment as physicians in our community, we have to do the work. We have to do those investigational trials. We have to do that investigational and comparative research to know, well, if I give a patient a drug that has systemic side effects and that not everyone tolerates, is it safer? Is it more efficacious? Is it potentially cheaper to apply a human cell and tissue product early in a process of missing, damaged, degenerated, deranged structural tissue and apply that biologic as an allograft, and by way of objective measures like nerve conduction velocity and monofilament testing, be able to prove if that's efficacious. Again, this is all theoretical, but with potentially 30 million patients in the United States suffering from neuropathy, is the burden of disease significant enough to make it much less than theoretical? I believe it is. And I hope that some of those guardrails are removed in the upcoming years so that those of us that are dedicated to the science, those of us that are invested in patient outcomes, can do the hard work, can do the investigational research, can do the top shelf clinical studies, randomized, controlled, comparative, so that we have some more answers for our patients. And ultimately, we have some more solutions for our patients. Have a very blessed day, folks.